Hello everyone and welcome to a special parasitology seminar. My name is Abhijit Chaidi, so I'm pleased to be your moderators of today's session. So today it is our pleasure to have Dr. Pipat Piewngam as invited speaker. But before we start, I would like to state that this seminar is organized by Department of Parasitology, Konkan University. All contents and discussion in this session is for educational purpose only. And it is my pleasure to introduce you about Dr. Pipat. Dr. Pipat currently he is a research fellow at the National Institute of Allergy and Infectious Diseases in National Institute of Health, United States of America. He received his PhD in medical microbiology in 2018 from Faculty of Medicine Sierra Hospital, Mahidol University, under the GJ Scholar. From this scholar, he got published in the research in the Nature Journal, which is the one of the highest ranked journals. And he also awarded as the National Research Award from his PhD dissertation from the National Research Council of Thailand in 2021. So today he will share his own experience on the research in the topic journals of a young scientist, how to succeed in publishing in the Good Impact Journal. So please join me and welcome Dr. Pipat Piewngam.
All right. So thank you so much for the nice introduction. So today, like Ajahn said that I will talk about the, so first when I was invited, like, okay, you like, please give some talk about how to publish in high impact journal. So, but at the beginning, I'm like, I have to be humble because I think this is really, interesting topic. So how to publish in high impact journal. So even me right now, so I keep asking this question all the time, So today it's just like, I want to share my experience instead of, telling you how to publish in high impact journal. And like I said, I want to be like humble a little bit. So I will change a topic a little bit. So today I will talk about a journey of the young scientist, how to success in publishing in good, high impact journal. So let's start with this first. What is the impact factors? So when we're talking about the impact journal or impact factors, so meaning that is the number of citations that received a year, basically like when you publish, you get more citations and that journal, will have the number of the total citations, and those numbers, reflect the impact factors. So the more you get citations, the more impact factor you will get. And then when we're talking about the high impact journal, so what is really high impact journal, high impact factor journal? So most of the people, when they say, oh, high impact factor journal, so you will think about Nature, Science, Lancet, JAMA, Cell, or New England Journal of Medicine, for example. But so when we're talking about the high impact journal, you cannot really compare, pear and apple or orange and apple. So I do search a little bit, so you have to compare the things in the same discipline. For example, if you do research in the blood research, for example, you have to compare within the same rank or within the same discipline that they also research the blood, for example, and what is the real impact factors. So the high impact factor journal doesn't mean that it's a good publications, and again, so the low impact factor journal doesn't mean that that is the bad publication. So just keep in mind that the high impact factor journal always have a lot of trash, I would say, and again, so the low impact journal also have a lot of good publications. So the high impact journal doesn't mean that it's a good journal. So, all right, then how to get published in good impact journal. So from now on, I will use good impact journal instead of high impact journal. So first of all, everything takes time. So please keep in mind that, if you want to publish something in good impact journal, so you cannot rush about the research because everything takes time. I will tell you how it takes time. So the second of all, the quantity does not equal quality. you know, a lot of researchers, they like publish 10 papers a year, you know, but if you compare the things in that paper, in those papers, so you will see maybe sometimes, yes, they have a lot of quality, they have a lot of quantity, but if you look deeply, maybe it's not a good quality. So then quantity does not equal quality. Sometimes, you know, for example, like I said, you know, back to the first thing, everything takes time. So sometimes one paper you take like 3 years, for example. So I will give you some example later. And again, so if you want to get published in a good impact journal, so the first thing that a lot of journal asks you is that is novelty enough? So what is the novelty? So meaning that this paper, this publication didn't, you know, the main results and conclusion is not, has been published or submitted somewhere else before. So it has to be new. But how you say that this is new, we will talk about that later. It's interest. Is it really the outstanding scientific importance? So for example, if right now you see a lot of people talking about the COVID-19, right? So like right now the interest is, you know, go to the COVID-19, but it doesn't mean that if you do research on COVID-19, you have a chance to get published in good impact journal, because the first one, is it novelty enough? Even you do the same interest, you have the same interest, you have things in COVID-19, for example, but those things, if it's not novelty enough, so you don't have a chance to get published in good impact journal. Oops. And the last one, so the conclusion of the interest is, you know, lead to the leadership in that field or not. Like, for example, when you read some paper, you will feel that, oh, this is really interesting, this is really useful, you know, the same things. If you have your own paper, you have to read that first, and, you know, you will conclude by yourself that this is interested enough or not, and this is will bring a lot of, you know, attention from the audience or not. Okay, so when we're talking about the novelty, so you know, the new story, let's say, how to get the novelty story. The first one, do a good research. You know, of course, you know, even you do some is not novelty enough, but you still have to do the good research. The second one, you have to read paper, you know, from the master, or read a lot of you know, literature review, or this some publication in your field, to see whether if this thing has already been completed before, you know. So it takes time, so you have to read a lot of papers. And so the third one, keep your curiosity and keep asking questions. So this is really important. So whenever I you know, teach my student, I'm like, being a good scientist, or, you know, being a good doctor, you have to keep the curiosity, and you have to keep asking the questions. So you have to like, why is like it? So what is that? What's next? For example, you know, keep the curiosity all the time and keep asking the questions, and those things will bring the novelty and, you know, reach the good research or good publication. And again, so thinking outside of the box. You don't have to thinking the things in the box all the time. You know, try to think about what else, if, you know, if you're thinking outside of the box, what will bring the new things or not, something like that. So I will, you know, give you some example about this thing later. So when people ask me like, who is your idol? So I always say that Alexander Fleming is my idol, you know. So Alexander Fleming, he discovered the penicillin, right? But he discovered the penicillin by accident. So Alexander Fleming, he's working with the Staphylococcus species, and then, you know, he played the things, the culture on the plate, and then just leave it like that. And then he took vacations. And I believe after his vacation, he said, oh, wait a minute. he looked on his plate on the bench, and he sees something like this, the huge colony. And later on, he characterized, and then he said that this is penicillium's colony that produced penicillin against Staphylococcus species. So because of Alexander Fleming, he keep his curiosity. Like I told you beginning, for example, you have to keep your curiosity for all the time. Sometimes if you don't have any curiosity, so you see this plate, okay, this is not really interesting, and then you trash it. So you miss the big thing, right? And again, so he get published in the British Journal Experiment of Pathology. So the impact factor about 1.7. So you see, this is low impact journal, but bring a good discovery. Like I said, a good high impact journal doesn't mean that it's a good journal, and low impact journal factor doesn't mean that that is a bad publication. So you have to keep this in mind all the time. All right. So let's talk about my story a little bit. So I'm working with the gram-positive bacteria, to see the interaction between gram-negative bacterial species, the interspecies, and also, the interaction between host and microbe. So back to, I think, 2015, I have my first hypothesis. So I want to see whether if people who have Staph aureus in the gut or in the nose, they have any difference in terms of microbiome or not. So I start by collecting the sample, the feces samples and the nasal swab, from the Thai population all over, Thai regions, north, northeast, central, and also the south. So I got 200 people who were the healthy volunteer, and then I swab the things, and I get the feces and I culture them. So we get about 99 people, 25 people who have Staph aureus in the nose and 26 people who have Staph aureus in the gut. And then I isolate the DNA to, study the microbiome. So when I got the result from the microbiome studies, as you see in this figure, so they have no difference in terms of microbiome profile. People who have Staph aureus in the gut and people who don't have Staph aureus in the gut have the same microbiome. So this result doesn't fit my hypothesis. What else we have to do? What's next? So maybe if you don't have any things support after this, so this project is done, right? Because you don't have anything to do after it, because that this result doesn't fit your hypothesis. So you don't know what you're gonna do next. But when I culture this sample, for example, I did my culture the fecal sample on the plate. The Staph aureus can grow on the metatarsal agar. The plate start with the pink plate. If Staph grow on this plate, you will see the yellow colony. And again, so when I saw this plate, I keep, because of my curiosity, so I keep asking questions that why people who don't have Staph aureus in the gut, they have a weird colony in this plate. If you see the difference, right? So the Staph aureus colonizer, obviously they have Staph aureus on the plate. All over the plate is Staph aureus. But Staph aureus non-colonizer, they don't have Staph aureus on the plate, but they have something really interesting on the plate. And later on, after the extensive experiment, we found that this is Bacillus species. Later on is Bacillus subtilis. After I saw this plate, I keep asking my mentor, both in Thailand and also in America, like, what do you think about this? And both of them said that it's normal. Bacillus produce some bacteriocin, Bacillus produce some antibiotic. People knows already that they can produce antibiotic, they can produce the bacteriocin. But again, so I don't believe that. I think it has to be something. And after this plate, after we know that this is Bacillus species, so I did some co-culture. So I used the Bacillus co-culture with Staph aureus to see whether if Bacillus can kill the Staph aureus, right? So because people say that yes, Bacillus can produce a lot of antibiotic, Bacillus can produce of bacteriosis. So maybe Bacillus can kill the Staph aureus. But again, so after I did that experiment, Bacillus cannot kill the Staph aureus. This is the different story already. So first of all, the microbiome profile between Staph aureus colonizer and Staph aureus non-colonizer is different. Is no difference. And then Bacillus on the plate of the Staph aureus non-colonizer cannot kill the Staph aureus, you know. And then what's next? So because of this thing, you know, we have, you know, extensive experiment to find out what's going on. And then we found that Staph aureus use quorum sensing system. You know, quorum sensing system is the system that bacteria interact each other, like they talk each other. So I always say that bacterial quorum sensing system is the bacterial Facebook because they used to communicate each other. So, and then we found that Staph aureus use quorum sensing system for the gut colonization. And Bacillus subtilis can produce some substance. So later on, we found that Bacillus subtilis can produce fungicin. And this fungicin substance can interfere the quorum sensing system. And then, you know, after we give the Bacillus spore, they can diminish the Staph aureus colonization. So, and we get published in Nature. So this publication took almost 4 years. See, if you want something big, so it's like, it's really take time. So for example, if you want to publish this thing real quick, you can publish within a year, but not for the whole story, right? So you can publish like, okay, Staph aureus use quorum sensing system for the gut colonization. That is the one story already. So you can publish within a year. But if you want something big, if you want to publish something in the good impact journal, it takes time. You have to plan really good experiment and then close all the gaps and try it. And I will give some example later. So after we publish this thing, I keep asking the question like, oh, can we really apply this story to other bacteria? Sorry. Okay. So what about if we apply the same story but different bacteria? For example, Staph aureus, the gram-positive bacteria, they have the quorum sensing system. And also, what about the other gram-positive bacteria? For example, Enterococcus faecalis. Enterococcus faecalis also have the quorum sensing system. When you compare these bacteria together, you will see that the quorum sensing system of two bacteria really, have this similar pattern, have the similar system. And then I'm like, okay, maybe we can apply the story that we discovered before with the E. faecalis. So after I tried to, use the same story with the different bacteria. So the first question is that if E. faecalis used quorum sensing system for the gut colonization, because we found that Staph aureus use quorum sensing system for the gut colonization. And what about the E. faecalis? Because they have the same, same system. And I found that the E. faecalis don't use the quorum sensing system for the gut colonization. So this is done. So same story cannot apply to the different bacteria. And again, so we didn't stop with the result. After we got, negative result, we keep asking question that, okay, and it has to be something. It has to be something. So what's next? Again, so after I read a lot of papers, so a lot of the Enterococcus faecalis, a lot of patients got this thing, this bacteria, and then have the bacteremia after, especially the immunocompromised patient. And then I keep asking question that, okay, what about if Enterococcus faecalis use quorum system for bacteremia? And yes, so the question, the answer is yes. Enterococcus faecalis use quorum system, to cause bacteremia. And how they can cause that thing. So faecalis colonize in the gut, and then they destroy the gut epithelial cell, and then go to the bloodstream, cause the bacteremia, and then after extensive experiment, we found that Enterococcus faecalis use their quorum system, and one thing is that they produce the protease and gelatinase, and those enzymes break the intestinal epithelial cell and allow them, from the gut, go to the bloodstream and cause the bacteremia. And again, so we got something really new story already. So different bacteria, different bacteria, similar system, give you different story. And then we go back to the same story about probiotic bacillus, because from the first discovery, we found that Bacillus subtilis can produce fungicide that interfere the quorum system of Staph aureus and diminish the gut colonization. And what about if we apply same thing, to the E. faecalis? Bacillus can diminish or can prevent the bacteremia or not? And we found that yes, probiotic bacillus can prevent the enterococcal bacteremia by produce the either fungicide or surfactant, the same substance, and those substances can interfere enterococcus quorum system and prevent bacteremia. This is example that how to get the new novel story, how to create the idea. Again, so you have to read a lot of paper. I would say read a paper, maybe not a lot of paper, but you have to keep reading a paper, and you have to keep asking the question, keep your curiosity. And and after you get the negative result, don't give up. You have to have the plan B, plan C, or sometimes you have to have plan Z, for example, to get something new. And recently I found something really interesting. When we do the histopathology, we always want to see the difference between control mice and infected mice in terms of the gut morphology. This figure shows the gut morphology of the infected mice between bacteria A and bacteria B. So after I get this result, I'm like, okay, maybe this is not different, because if you can see that they have the beautiful morphology, you have the beautiful pattern of the histopathology in the gut, right, between A and B. But thinking outside of the box, maybe they have something different in terms of mouse A and mouse B. And if you look closely, you will see something different. For example, mouse A, you will see that the goblet cell number of mouse A is really, really low compared to mouse B, right? So this is like when I first get those two figures and I compare, I'm like, okay, this is maybe it's already done because nothing different. But when you have time, it takes, like I told you, everything takes time. You look closely to this result and you're like, okay, this is something different. So the mouse the mice infected, the mouse A, they have less goblet cell, but the mouse B, they have normal, let's say normal or high number of goblet cell, and this bring the new thing. So basically, this project is going on. So hopefully we can get it done soon. So this is the example of the best thing of the curiosity, or the best thing of asking a question, and the best thing of you're reading a paper, so you have an idea that so what is different, what is new, what is old, what is already has been done already, for example. So those things, come from the best experimental plan. And how to plan an experiment. My mentor always said that when you plan an experiment, you have to think about it. This is the skeleton system. You have the main idea, you have the main result as a core, right? So this is the core result. And then you have to plan the experiment to have the supporting evidence. And that is the organs. So you add your arms, you add your legs, you add your head. So, and then you assemble as the skeleton system. But do not plan experiment that you have too many topics in one paper. Don't do this. So you have to have only one main message, and then that main message is a core experiment and core result in your publication. And you keep adding the supporting evidence, to complete this thing. But whenever you add more evidence, you add more story in the same paper. So for example, you found that bacteria A used this enzyme to break the epithelial cell, and you want to add more that, okay, maybe bacteria A used those host immune system, activate host immune system that lead to the epithelial cell breakup. Those things have two different stories already. So the first thing is bacteria use their own system, right? And you add more that bacteria use the host system. So my mentor always said that don't do this, because the more you add different story, the more you will get the feedback from the reviewer, and that is destroy yourself, and that is destroy your story. If you read the paper from whatever, Nature, Cell Science, you will see that they only have the core experiment, and they have just, try to add more experiment to close the gap in the same direction, but they never have 3 stories in one paper, 3 different stories in one paper. Don't do that, because you choose yourself already. So you're not gonna get, published in the high impact journal if you do that. So this is an example that how you, plan a good experimental plan. How to have the experimental plan. So after you've done your research, and you're ready to submit your publication, you're ready to submit your manuscript to the, some high impact journal, you have to ask these questions first. Have you done something new? Again, so you have to read the paper first, right? A lot of paper like this, the thing that you just discovered, is it new or not? And then, is there anything challenging in your work? Is it really difficult, or you discover something new that's really challenging? And this result may be influenced to other researcher. So if, for example, if you read your own paper, do you want to follow this thing or not? You want to repeat this research or not? Or you think that this is really useful for your research or not? And the last one, if this thing really, provide the solution for some difficult problems, for example, some, MDR infection right now, if this thing really solve this problem, for example, if you discover some new antibiotic, this new antibiotic can, solve the same problem or solve the current problems or not. If you can answer these questions, keep in mind that you have a chance to get published in a good impact journal. So whenever, again, based on my experience, whenever I have the result, and then every Monday in our lab, we have a lab meeting. I present my result. My mentor keep asking that, what's next? So what's like, when I show them result, they're like, so what is exciting enough? If you cannot answer this thing, again, so the audience will feel the same when they read your paper. So try to, answer these questions before submit your manuscript. Sometime after you finish your manuscript, after you finish your experiment, let's say most of the time, you have to add more experiment to close the gap, because after you read your manuscript, let's say you read it 2 times, you will find the gap, and then you have to close the gap before you submit your manuscript. So after you get manuscript, you're ready to submit. You have to ask yourself next, is this manuscript because worthy? How to make this thing really worth it. So first thing, you have to have clear scientific message. Like I said, when you plan the experiment, you have to have the core result, the core research, and then you add the supporting evidence to close out the gap. But you cannot have the different story in the same paper, because that will open more gap, open more thing that reviewer will see those things. Remember that when the journal send your paper to review, at least you have 3 or 4 reviewers. I will tell you later like what is my experience. So the second one, the title and abstract. You have to have the catchy title. Let's say really catchy, like if you see this paper, if you see this title, you really want to download this paper and read it. So the same thing, you have to set the title and abstract that really catchy and really draw attention from the reader or from the audience. And what is a story? Again, so is it new or is it really interesting story? These things you have to have excellent, let's say excellent, the selling experience. Because when you publish the paper, like you try to sell the things, to the journal, right? You try to sell your story to the journal. So, and then you have to you have to sell the best, by writing the good story, by writing the good scientific manuscript. What should be included in the title? The first one, only one key message. When you read a lot of paper, for example, again, so from Nature Cell Science, you see that the title is really short, So basically, try to keep your title short. Don't use more than 10 or 12 words. And those 10 or 12 words have one key message. And if it's possible, try to condense the paper into the sentence summarizing the finding. And, and those things will capture the reader's attention. And when you see it, you really want to download and, read the paper. And again, what you should include in your title, try to use the action verbs to make it lively and keep it short, not more than 10 or 12 words. And don't use the generic words. For example, this study of blah, 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 the investigation of blah, 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 or this study on the blah, blah, blah. So don't use that thing. Or don't use the question mark or play on words. So the only things you can play on the words, or you can use a question mark on the title, is the review paper. If you're writing a review paper, yes, it's really catchy if you have a title, the question marks on your title, or you can play on words. But don't use this thing with the research article. All right. So after you send the paper to the journal, so the first thing you have to select the journal first, right? That journal really fit your research or not, that journal really fit your finding or not. The first thing, don't underestimate. I see a lot of paper that they underestimate themselves and they get in, let's say, low impact journal. Actually they can go higher. Then the first thing, don't underestimate your research. And the second one is, don't overestimate your research. For example, you have only 3 results and you said, okay, these 3 results is really fit for Nature journal. Maybe sometimes you overestimate yourself and then you lost a lot of time, because after you send it the paper, you need at least 2 or 3 months to get the feedback from the reviewer if the editor sent for review, All right, get back to this topic, decision from the journal. The first one, after you submit your journal, after you submit the paper, you will get, the first thing you might get is the desk rejection. What is desk rejection? Meaning that the editor don't like it. They don't send out for review, so they reject immediately. So usually the high impact journal, they will reject within half a day or at least 3 or 5 days, for example. This thing really quick, and more than 50% of the paper will get this stage. So most of the paper will get this rejection. All right, but if you don't get this rejection, meaning that editor already like your story, so they send out for review. This will take about 2 weeks, or sometimes take like a month or 2 months, depends on the journal. But most of the high impact journal, they allow reviewer to read the paper only 2 weeks. After they finish the review, you will get 3 things. The first thing is minor revision. Just send some word. Just correct some word. Change some sentence, and then it's ready to be accepted. But for the high impact journal, this thing really, really rare. You never, based on my experience or things in the lab, in my lab, we never get minor revision after we, first submit first round of review. But most of the paper, after they send out for review, you will get major revision. So major revision is that editor like it, reviewer like it, but they need a lot of things. They see a lot of gaps. And for example, recently I got one major revision for my paper from the 4 reviewers. Basically, they asked me to repeat all the mouse experiment. This is really major revision, and then you have to decide whether you want to revise or you want to decline. And this is really sad one. So rejection. So rejection is that editor doesn't like it, and also reviewer doesn't like it either, and then they reject it. So most of the rejection letter will say that this is not novelty enough. Maybe it has been published somewhere, and also it's not interesting enough, for example. And the last one is accepted. So the high impact journal is less than 10%. For some journal is like less than 5%. They will accept the paper, because so nowadays the high impact journal, high impact factor journal, they still have the book of the journal, right? So they have the limited space, and then they have to select the best one to get published in their journal. That's why you see that only like 10 or 5% get published in high impact journal. The first thing after you submit the paper, you have to write the cover letter to the editor-in-chief, to the editor of the journal. So this thing, the cover letter, will you try to, tell the editor that how nice of your story. The first thing, you have to address the editor by the name. You have to use the right name. Write to the right name. And try to avoid making a mistake in the name of editor and the journal. Sometimes you get rejection from the first journal, and then you want to submit to the second journal, but sometimes you forget to change the editor, the name of the editor, or you forgot to change the name of the journal. So this cover letter, you have to make sure that it's not too short, or it's not too long. And again, you cannot repeat the abstract. So this has to be something new in this letter. So this letter, you have to highlight the novel aspect of your work, and to tell the editor that why the journal would find is really important. Because if you don't have this cover letter, so sometimes the editor don't have time to read the paper, and they don't know that what is the novel story in this manuscript. So that's why you have to write a good cover letter to let the editor know that this is the new story, this is the really interesting story, and this is really worth it if we publish in your journal, for example. And yeah, so indicate why this work fits the journal scope or the journal merit, for example. So after they send out for review and then you get the major decision. So what's next? People will like panic. So don't panic. But it's really easy to say. But again, so don't panic. Try to read the comments twice or more, and try to address the comments fully and honestly. Try to provide answer all the data for all comments. So like I said, for my paper, so we took like 3 years to revise all the things that reviewer asked us to do. So we have to provide all the answer to all the data that they comments to our paper. And again, this thing will come with the good experimental plan, a good idea, and again, you have to have your curiosity. And this thing is very sad to say, but it's real. So after you get the major revision decision, you always apologize for some mistake or some misunderstand, even they are wrong. So sometimes when I read the comment from the reviewer, I'm like, this is wrong. So they are wrong. They don't understand our story. But again, so when you write the rebuttal letter to tell them that, oh, this is the right story, but you have to start with the apology, for example. So if you cannot do some experiment that they requested, so just tell them that why you don't do this experiment or why you cannot do this experiment. So first of all, try to answer as much as possible. But if you can't, just tell them that why you cannot do this. And what about if you get a rejection letter? Don't panic. So read the comments twice or more. And you can appeal it. So if you think that your story is really good enough and you think that this is not fair, so reviewer don't understand your story, this is, they judge your story not fair enough, you can appeal this thing to the journal. And maybe the journal will send back to reviewer and you have a chance to get published in that journal. And this thing, we always do this, based on my experience. If we get one rejection from the first journal, so after you get rejection, they will give you the comments and you will read all the comments and you try to perform all the experiment that reviewer suggests you, and that will bring your story really strong. And then it's ready to submit to the new good journal. So it's really rare that like, if you submit your paper and then you will get published right away, it's really rare. So it takes time, like 2, 3, or 4 years. For some paper, some big paper, I found that some story they took 7 years, for example, to get published, and those process. So keep in mind, it takes time. And again, so I keep telling you that, okay, you have to keep your curiosity, you have to keep asking the question, you have to keep reading the paper. But again, so we are not a robot. So you have to have work-life balance. You have to refresh your brain, you have to refresh your mind all the time. So then if you have things that stuck in your brain, you have things that stuck in your mind, you're not going to have the good idea, you're not going to have a good mood to plan your experiment. So easy, just work-life balance, do the 6 things that I just show you in this slide. Try to add more exercise, take your vacation, or prioritize your job, that what is should do first or what is can be wait, for example. So work-life balance will bring the things that refresh your mind, refresh your brain, and will bring your good curiosity, and you can have a good idea to make your research better. All right, I think it's time for the summary. So the successful publication of the work in a good or high impact journal start with choosing the important question, right? That thing is really interesting right now or not. It's really solve the problem. What is the real problem right now? Try to answer all the questions. Try to choose the thing that you can answer the important question. Design a sound study and statistical power. You have to have the to add more evidence to close the gap, but don't have try to avoid to add more story. Performing the work with the impeccable integrity and attention to the detail, and also this thing, writing an excellent manuscript, try to save your stuff and submit to the right journal. Don't overestimate, and again, don't underestimate. You have to choose the right journal that fit your story. And try to respond to reviewer comments fully after you get the feedback. And don't forget to have the work-life balance. This is really important, to refresh your brain and then to refresh your mind. All right, so I would like to close this story with this slide. Dr. Shinya, the Nobel Prize in Physiology and Medicine in 2012, he said that I can't see any failure as a chance. Remember, after you get your negative result, don't give up. Try to see that it's a chance to have a great story later. All right, thank you.
Okay, thank you very much, Dr. Pipat, for a very informative talk. So we are in the Q&A session. So if anyone have a question, you can turn on your microphone and ask the question, or the typing in the chat box also welcome.
Ajahn Apisit, may I have a question, please?
Yes, Ajahn.
Thank you, Ajahn
Pipat.
Pipat. I try to get your name. Thank you very much for your talk. Very nice talk, actually. First of all, I would like to thank you for your presentation. That's for our seminar series. We would like to open the seminar series for our students to get successful and well-known, like world-class one. And one of our staff recommend your name. I never know you before, but I see your publication was very impressive. So I think this is a good choice that bring you to be a presenter for our student to get the idea of how to get the high impact factor publication. And I like your title, Young Scientists to Publish in High Impact Factor. Because, when I'm talking to the student that we need to publish in a high impact factor, people would say, yeah, you are senior, you have a lot of things, you have experience, you have a lot of property to do that, but I'm young, what to do that? And I think you open the world of, how young scientists to get this idea to get the high impact factor. And I think you are really, I'm really thank you, because I know that in DC it's now like close to midnight, right? Very late at night.
Yes.
Okay. I actually do not have the questions, but I have a comment like I already told you, and I think I would like you to explain a little bit more. You already told us how to get a successful and what the idea of to get the good high impact factor publication. I think the most keyword that you give to us is the student have to do hard work, good research, and novelty. So they need to clarify if the research that they have done is novelty enough to get that. But the most important words that you gave to the student that I really like, and I would like the student to take this word, the student have to keep reading, update all the time. I think this is the key successful for the student. I think students have to read a lot of, update publication like that. So back to my question. So from my experience, you are young. I think you are less than 40 years, maybe you early 30 or something, and you published in a very high impact factor. You got a very nice job. And can you tell us a little bit more that how you get to work in NIH? From your, you finished PhD from Thailand, and then I think you were successful to go to work with the good international firm like NIS.
So okay, yeah. So thank you so much, Ajahn, for the things that you just said. So I hope this thing, answer the question that how to succeed in published in the high impact journal, like you said. And then, I hope that this can be, have some inspiration for the students, at the Khon University. All right. So back to like when I was the PhD student, my mentor, he went to Thailand, and then so we have some seminar, like a special seminar in my department. And then I talked to him. I told him that, okay, I have this idea. I have this story. I want to work with you. If this thing is possible or not, and then we just, develop the idea, develop the research that is really fit for him and also fit for me. So I would like to say that if you want to work with the, for example, NIH, at least in from experience in my lab, so people will ask, what is the win-win situation? So if you work for him, so what he gonna get, and what you gonna get. So this is like, you try to find a win-win situation. So for me, I quite lucky that so my mentor is like he went back to Thailand, and we have a chance to have some scientific talk, and then we develop a proposal, and then I can I go to the NIH. But my first year as PhD student at the NIH in US, right? So because of this thing, so I try to, my first year as PhD student, I try to come up with the great idea, come up with the, not the great, sorry, come up with an idea all the time that, oh, this research has to be like this research has to be like that. So this is new, this is, is really novel. So I keep this thing a lot. And, my first year, I keep telling this thing to my boss, to my mentor. And he see that, oh, we have a chance to work together for the long period. So, and after my first year, so he just like, oh, if you want to stay here as the postdoc, so we can do that. So that's why, after I think 2016, from that, I just keep working at the NIH at the postdoc, and I just got promoted this year to be a research fellow. So again, so you have to be ready to, see the world. You have to, because when you work abroad, for example, NIH, they don't have time to train you. You have to be ready to work with the new things, right? So that thing come from reading, understanding, or, like I said, I love to use this word that your curiosity, your curiosity will bring you some reward.
So when you did your PhD, you have done your lab work in NIS as well?
So my PhD thesis, I have two parts, like two big parts. So one part I did in Thailand, and yes, second part I did at the NIH.
Okay, so you have experience in NIS before while you did your PhD.
Right.
So we have questions in the chat box, I think, from Solatan. He asks that, is there a certain time limit for the research that published in journal? For example, the research that completed in 2018 and would like to be published in 2021. Is there a time limit or suggestion for how long it should be? Just need a chat box probably better.
Yeah. Is there a certain time limit of research that published in the journal is the research completed? No. So it doesn't have to be. It doesn't matter. So if you finish your story in 2018 and you wanna submit your journal, you wanna submit your paper in 2021, so you can do that. But from 2018 to 2021, maybe your story is old already. So someone will get published before, and then you don't have a chance to get in a good, impact journal. So I don't think you need to wait. I don't know why you need to, wait until 2021, for example. After you finish your story, write a paper and then submit right away. Not because like I told you, so it takes time. So you finish your story in 2018, you're not gonna publish in 2018 for sure, for the big journal, for example. So you need at least a year to revise the paper or to resubmit to another journal, for example.
Somchai. First of all, I would like to thank you for informative talk and congratulations for your successful. Even you are a young scientist, right? According to Ajahn Thevarat question, may ask at the beginning how to connection with the math overseas advisor, right? Because the selection of overseas advisor is very important. Even we see the international publication or well-known professor, but after that they have many problems. And for you are the first selection, what is the tick for your select? You would like to do with this professor? Very important.
Okay. Yes. Thank you so much, Ajahn. So this question I got all the time, like how you connect your things to the professor outside or the professor abroad. And I always answer that once you read a paper in your field, you know that who is the big names in your field, right? And then you go to Twitter, and I would like to tell a new people or a lot of people that so try to use Twitter, because so right now a lot of the professor, a lot of PI, principal investigator, they use Twitter to announce the job or show the story, show the paper, and then you have a chance to have a close contact to them. And again, so if they don't have a Twitter, if you don't have Twitter, just simply send them an email, to tell them that how you impress their story and why you need to learn more from them, for example. So this thing is really important. So besides my boss, I always send an email to like the big name in the US, for example. One of my paper, because in our lab we did Staph aureus for I think 20 years already, and the only one in the lab that who research in new bacteria, that is E. faecalis. So my boss doesn't have any experience in E. faecalis. They don't have any E. faecalis strain in our lab. So what next you have to do? You have to know, like I said, so once you read the paper, you know who is the big name, and then you send them an email, say that you impressed their your paper. This paper may be really useful for your story, maybe useful for your research, and you ask them for some help. And sometimes you can ask them for like if you have a chance to work with them or not. If they have like a position available, if you can use your profile that really fit what they're looking for example. So try to connect by either email, Twitter, Facebook, use this thing as the academic purpose.
Thank you a lot. And the second one, as you said, when we would like to submit our manuscript, like don't over or underestimate. And in this is very difficult for this situation, because you submit in Nature. How to select this is not over or not under. How to adjust for this, because we would like to submit, we can find the journal right in the many websites, but for you how to adjust for this?
Right. So like I said, so first of all, you have to read a lot of papers. So for example, Nature. after you read a lot of paper in Nature, you will see the theme. I would like to say the theme that what they are looking for. You will see the same pattern that published in Nature, and then you try to compare to your story whether this is the things that Nature looking for or not. I would like to say that if you have your great story, aim high. If you have a great story, aim high. I think you the only one know the story the best, that is the best story or not, right? So sometimes, like I said, people really underestimate their story and then to send to the low impact journal. So that is like a sad story that you have a chance to get in high impact journal, but sometimes you just want to publish real quick. You just lower your chance to get a high impact journal because, like I said, so it takes time. It's take at least 2 or 3 years to get in those journals. So you read more and then you will see what is the theme for those journals. For example, I just got rejection from New England Journal last week. So we did some trial in Thailand. And once we write the story and we see that this is really fit to the New England Journal, because first it's a trial and it's answer the public question that we use the probiotic bacillus to eliminate the Staph aureus colonization. So this is really a important question that we can treat them, because right now we have a lot of problem with the antibiotic. Staph aureus, they're resistant to a lot of antibiotic, and then we cannot treat them with antibiotic. So we try to find the new way to treat this thing. And after we finished the story, we said, okay, our story really fit to the New England Journal. So we sent the paper to the New England Journal, and it either like it, but sadly, they said that they don't have enough space to publish our stories. So that's why we get rejection. So we will try to another journal. So this just for example, I think I believe a good scientist never overestimate their story, but most of them just underestimate their story. So I would like to say that just try to submit to the high impact journal first, or sometimes it's a good thing, So after you submit to the high impact journal, you get the reviewer comments, even you get rejection, you know that what is they're looking for, and you try to finish the things that they just comment and submit to another high impact journal. I think you have a best chance to high impact journal. For example, one of my story, the e-weekly story. So first we submit to Nature Medicine. So we get a lot of comments from the reviewer, but they reject our paper. But we try to perform the experiment that they just request, and we send to the science paper, and then we get accepted in science in the science paper. So this thing, for example.
Thank you for your share your experience. And one more, I like this word that you suggest for the audience. You said work-life balance. I like this word very much, because when you success, you have the tick for work-life balance, right? Could you tell a little bit of your tick for our audience?
Right. So sometimes it's really hard to say that you have to be work-life balance if you have a lot of pressure. You want to finish your experiment, right? For example, let's say based on my experience, I have to finish one experiment. I keep repeating this experiment 50 times, 50. And after that, I'm like, no, this is not going to work like this, And then I just like go to the gym or, go to DC, go to the museum, to try to refresh your brain, try to refresh your mind. Because let's say if you already repeat them 50 times and it doesn't work, the 51 time, it's not going to work as well. You have to change something, and that thing is yourself. You have to change yourself first. Try to refresh the mind. And I always, if I have a lot of pressure, if I have a lot of stressful, I just go to the gym. That's I did all the time. Go to the gym, or go to the museum, go to DC, have some drink. That is like a really great work-life balance for me.
Thank you very much. Okay.
Okay, so any more questions? Okay.
No question from students?
I think I got one question in the box, right?
Yes.
Is it possible that a reviewer have their view contradict each other? If it's possible, that how many? Yes, so this thing we get all the time. Okay, I will repeat the question. So is it possible that the reviewers have their reviews contradict each other? If it's possible, then how to manage this situation? Yes, so this thing, like I said, you have like 3 or 4 reviewers, and some they contradict each other. The only thing is that just follow the thing, the one who just agree with your finding, and tell the another one that, so maybe you just misunderstood, or this thing is already correct. That thing, to manage this situation. So it's good to have, they contradict each other, instead of have 2 reviewers that they're like, oh, this is trash, for example. So this is a good thing. It's already that you have to contradict comments.
Okay.
I have one question. Nice to see you, Dr. Pipat. I'm a big fan you on your Facebook.
Oh, thank you so much.
I have one question. For the student, how to keep the passion for in a wrong in on the research. How you keep the passion, because
Okay.
I think you have a lot of the passion in a long time.
Right. So first of all, you have to ask yourself first that what are you doing right now is the right thing that you would like, to do in the long term or not. If you say yes, you just, because just how can I explain this thing? So for example, for me, I'm really excited all the time when I saw something new, when I see some different story, or different things that bacteria produce or bacteria behave with our body, for example. So I think how to keep the passion for the long term, I think the first thing, just ask simply ask yourself that do you love the thing that you do right now or not? So if the answer is no, so then just change it. But if the answer is yes, just keep doing it, don't give up.
Thank you.
May I share this? May I share this answer?
Yes, please.
I think you need to find what is your passion first. People have different passion to do the work. Some of them, do the work for, like Dr.Pipat, he loved to do science, he enjoyed to do research in the laboratory. So he enjoyed every time he found a new result. That is his passion. But some passion is not like me. My passion is I like to publish in a good journal, because that is my career. I need promotion, I need publication, and I want my work to go to the good publication. People read it and I become a famous. So that is my passion. So you need to find what is your passion, and you do it and you're happy. So if you're happy to publish, you do it. You're happy to do the research in the lab, you do it. So I think it's every people have different passion, but you need to find what is the way of your passion to success in your work, and then you're happy in your life. I think that's
Right.
the best way to get a
Yes.
successful.
Yes, I think this is the best question. Sorry, this is the best answer. Yeah, like you have to keep yourself like keep asking yourself that this is you love it or not, and what is the real passion that you would like to be.
Okay, so any question from student? So I think we don't have any more questions right now, but if student have question or others have question, you can emailing me or just tell me that you have a question to ask Dr. Pipat, and I will forward this question to him and reply and answer you again. So before we close the session, I would like to invite Professor Somchai again to express our gratitude on behalf of the department for Dr. Pipat, please.
On behalf of our program and our department, I would like to express my sincere thank you for your inspire of our graduate and our young lecturer, right? Because you are young scientist, but you can publish a high paper. As Ajahn Thevaraj said, only the famous professor or the famous or the senior one can publish a high impact journal like you, right? But you are young scientist, but you can do this. Therefore, you make inspiration for our graduate student, and you also tell the trick and many path that you success or your story to how to conduct or your high impact journal. I appreciate you a lot and hope that in the next time you will share your experience, right? And when you come back and don't forget to work in Thailand and forgot to Thailand, come to Thailand and work in anywhere in the university in Thailand for have connection and have I think in the future we will have a connection with our student or our new staff, or maybe we will have a connection during in USA also, right? And at this moment we didn't provide you some souvenir, right? How to provide him souvenir, Ajahn Am. Because usually invite speaker, we have some souvenir for you, right? We hope you will come here and you will give us some souvenir for our program for your memory. Thank you very much. Thanks.
Thank you so much.